The 1974 publication of Christian Duve's article Microbodies in the Living Cell in Scientific American represents a pivotal moment in the historical trajectory of cell biology. It is a work of synthesis, a narrative of discovery, and a bold postulation of function and evolutionary significance. This essay will undertake a profound and systematic evaluation of the specific internal claims, arguments, and conceptual frameworks presented within this seminal text. The analysis proceeds by dissecting the article's logical flow, assessing the strength of the evidence Duve marshals for each proposition, and examining the intellectual architecture he constructs to house a new and enigmatic organelle. The scope is an intimate critique of the work itself, exploring how Duve persuades his professional audience of the existence, identity, and importance of a previously unappreciated subcellular world. The objective is to illuminate the intricate process of scientific reasoning and conceptualization as captured in this document, a masterclass in transforming a biochemical anomaly into a fundamental component of the eukaryotic cell. Section 1: The Methodological Blueprint and the Urate Oxidase Anomaly. Christian Duve initiates his discourse by establishing the methodological foundation upon which his discoveries are built, a paradigm he terms biochemical cytology. He presents a compelling case for the power of analytical cell fractionation, a technique involving the homogenization of tissues and their subsequent separation into constituent parts through differential centrifugation. His evaluation of this method is direct. It allows the biochemist to perform a cellular autopsy, isolating organelles in sufficient quantity and purity for detailed enzymatic analysis. He recounts the successful application of this strategy in the prior elucidation of the lysosome, an organelle defined by its unique collection of acid hydrolases. This historical recounting serves a crucial rhetorical purpose. It validates the methodology before introducing the central puzzle. The core claim of this introductory section is that disciplined biochemical analysis of enzyme distribution among subcellular fractions can reveal the existence of new organelles. The work then pivots for the specific problem that seeded the discovery of microbodies, the curious behavior of the enzyme urate oxidase in rat liver homogenates. Duve meticulously details how this enzyme's sedimentation profile conformed to the pattern of neither the large mitochondria nor the smaller lysosomes. It settled with a distinct equilibrium density in the sucrose gradient, a clear indication that its localization was unique. The argument here is built upon the presentation of a clean, irrefutable anomaly. The evidence is purely biochemical, a set of data points on a graph that defy existing cellular maps. His claim is that this enzymatic outlier signals the presence of a distinct physical entity, a particle housing urate oxidase and, perhaps, other associated enzymes. Section 2: The coalescence of evidence and the christening of the microbody following the identification of the urate oxidase anomaly. Duve details the systematic search for other enzymes that shared its peculiar sedimentation behavior. This section's central argument is that consistent grouping of enzymes within a specific fraction provides powerful evidence for their colocalization within a single type of organelle. He presents the results of this search. Two other enzymes, amino acid oxidase and catalase, were found to purify with urate oxidase. The strength of this claim rests upon the convergence of multiple independent biochemical measurements, shared distribution of these three enzymes, each with a role in oxidative metabolism, suggested a functional linkage. Simultaneously, Duve evaluates the contribution of a parallel line of inquiry in electron microscopy. He references the work of cell morphologists who had identified small, single membrane-bound organelles with a dense granular matrix, sometimes containing a crystalloid core, in a variety of cell types. He makes the crucial associative leap, claiming that the biochemical entity isolated through centrifugation and this morphologically observed particle were one and the same. The evidence for this connection was correlational of fractions enriched in urate oxidase. Amino acid oxidase and catalase were also the fractions enriched in these small organelles. It was this correlation that led to the adoption of the provisional name microbody, a term of purely morphological description. The evaluation of this name reveals the state of knowledge at the time. It was functionally opaque, reflecting an entity identified by its physical properties of size and appearance, weighting a more profound biochemical identity. Duve's argument is a testament to the power of interdisciplinary synthesis, merging the quantitative precision of biochemistry with the visual acuity of microscopy to give substance to a new cellular component. Section 3. The formulation of the paroxysm concept. This portion of the article contains its most significant conceptual breakthrough. The central claim is that the colocalization of hydrogen peroxide producing oxidases and the hydrogen peroxide destroying enzyme, catalase, is the key to understanding the microbody's primary metabolic function. Duve proposes a specific and elegant biochemical framework. The metabolic sequence he postulates begins with an oxidase, such as urate oxidase or amino acid oxidase, which catalyzes the reaction of a substrate with molecular oxygen. This reaction yields an oxidized substrate and, critically, hydrogen peroxide. He presents hydrogen peroxide as a highly toxic cellular poison. The second step in his proposed pathway directly addresses this toxicity. The abundant catalase within the organelle then detoxifies the hydrogen peroxide through one of two mechanisms. The first is its classic catalytic activity, where two molecules of hydrogen peroxide are converted into two molecules of water and one molecule of molecular oxygen. The second is its peroxidatic activity, where catalase uses one molecule of hydrogen peroxide to oxidize another substrate, such as a phenol or alcohol, producing water in the process. This hypothesis provides a stunningly complete explanation for the paradoxical presence of enzymes that produce a toxin alongside an enzyme that neutralizes it. It frames the microbody as a specialized metabolic compartment for safely conducting oxidative reactions that generate hydrogen peroxide. Based on this powerful functional insight, Duve introduces a new name for the organelle, the paroxysm. He argues this name is superior to microbody because it defines the organelle by its unique biochemical activity, its central involvement in hydrogen peroxide metabolism. The evaluation of this conceptual leap is overwhelmingly positive. It transformed the microbody from a curious particle into a purposeful machine, solving a major biochemical riddle with a hypothesis of remarkable explanatory. Section 4. Expanding the family, the glycosystems in plant metabolism. Duve then extends his analysis beyond the animal kingdom, evaluating the discovery of related organelles in plants. The principal claim in the section is that the particles known as glycosystems, found in the fat-string tissues of germinating seeds, represent a specialized class of microbody. He credits the elucidation of their function largely to the work of Harry Beavers and his colleagues. The essay details the unique enzymatic machinery housed within these plant organelles. In addition to the characteristic paroxysmal enzymes like catalase, glycosystems contain the key enzymes of the glycosylate cycle. Duve explains the profound physiological importance of this pathway. During germination, before photosynthesis can begin, seedlings rely on stored fats for energy and for the building blocks of new structures. The glycosylate cycle provides the metabolic bridge to convert these stored lipids into carbohydrates, specifically sucrose, which can then be transported to the growing parts of the plant. His argument is one of unification. He asserts that glycosystems and animal paroxysms belong to a single, versatile family of organelles. The evidence for this claim is both morphological and biochemical. They share the same basic structure of a single limiting membrane enclosing a granular matrix. They also share a core set of enzymes involved in hydrogen peroxide metabolism. The key difference lies in the presence of the additional, highly specialized enzymes of the glycosylate cycle in the plant variant. The evaluation of this argument highlights Duve's capacity for synthesis, weaving disparate lines of research from animal and plant cell, biology into a cohesive conceptual tapestry, the microbody was becoming a universal feature of eukaryotic cells, a theme of fundamental importance with variations tailored to the specific metabolic needs of the organism. Section 5 Evaluating a primitive respiratory role. The penultimate section of the article moves into a more speculative and deeply theoretical domain, assessing the potential evolutionary origins and broader physiological role of paroxysms. The most ambitious claim Duve presents here is that paroxysmal metabolism represents a form of fossil respiration. He postulates that this organelle is an evolutionary relic, a remnant of a primitive metabolic system that predated the emergence of the far more efficient mitochondrial respiratory chain. His argument is based on an assessment of the paroxysm's bioenergetics. The oxidation of substrates in paroxysms, he explains, is directly coupled to the reduction of molecular oxygen to water via the hydrogen peroxide intermediate. This process, unlike mitochondrial respiration, is a thermodynamic dead end. The substantial free energy released during the oxidation is dissipated entirely as heat, with no mechanism for conservation in the form of adenosine triphosphate, or ATP. He presents this inefficiency as a hallmark of a primitive system. The paroxysm, in this view, was a necessary innovation in an early, oxygenating atmosphere, allowing primordial cells to carry out essential oxidations and detoxify oxygen, a potent poison to early life. It was later superseded by the evolution of mitochondria, which harnessed the power of oxidative reactions to generate vast quantities of chemical energy. The evidence for this evolutionary hypothesis is necessarily indirect, built upon logical inference and comparative biochemistry rather than direct fossil records. He supports it by framing the paroxysm as providing a metabolic overflow or auxiliary system in modern cells, handling specific oxidative tasks for which mitochondrial pathways are ill-suited. It is a bold, almost romantic, piece of scientific speculation, one that provides a grand evolutionary narrative for his newly defined organelle. Section 6 The question of biogenesis and cellular integration. In his final substantive arguments, Duve addresses the critical question of how paroxysms are made and maintained within the cell, a process known as biogenesis. At the time of writing, the prevailing models for organelle credation were mitochondrial and chloroplast division, or the budding mechanisms associated with the Golgi apparatus and lysosomes. Duve evaluates the evidence pertaining to paroxysms and finds it points in a different direction. His claim is that paroxysms arise as outgrowths, or buds, from the smooth endoplasmic reticulum. The evidence he cites is primarily morphological, based on electron micrographs, that show apparent direct physical continuities between the membrane of the endoplasmic reticulum and the membrane of developing paroxysms. He interprets these images as snapshots of an assembly process, where the organelle's membrane is derived from the extensive network of the endoplasmic reticulum, and its enzymatic contents are synthesized on ribosomes and subsequently imported. This claim was remarkably prescient, while the full complexity of paroxysmal biogenesis, involving both the endoplasmic reticulum and the import of proteins from the cytoplasm, would take decades more to fully elucidate. His core hypothesis of an endoplasmic reticulum connection set the research agenda for years to come. The evaluation of this specific claim underscores his keen ability to interpret morphological data in a dynamic, process-oriented framework. He was thinking about cell as a living, manufacturing system, as most mechanism for paroxysm formation and logical extension of that world, integrating the new organelle into the cell's existing endomembrane system. In conclusion, Duve's Microbodies in the Living Cell stands as a monumental work of scientific exposition. A detailed evaluation of its internal claims reveals a carefully constructed argument that progresses with inexorable logic from a puzzling biochemical observation to a complete, functionally and evolutionarily contextualized cellular organelle. The work's enduring strength is rooted in its demonstration of a powerful research paradigm, the fusion of quantitative biochemical fractionation with high-resolution microscopy. Duve's central claims regarding the existence of a unique particle defined by a specific suite of oxidative enzymes, the elucidation of its core hydrogen peroxide-based metabolism, and the conceptual unification of animal peroxisomes and plant glyoxysomes have all been substantiated and are now foundational tenets of cell biology as more speculative but deeply insightful hypotheses concerning the organelle's primitive respiratory function and its biogenetic origin from the endoplasmic reticulum, while subject to refinement and immense subsequent research, established the intellectual framework for future investigation. The 1974 article is more than a simple report of discovery. It is a profound articulation of a way of seeing the cell, a narrative that persuasively establishes the peroxisom's identity and secures its permanent place in the intricate cartography of the living cell.